This is the first of 4 lectures for Unit 6. This section is going to cover the diverse array of different organisms included in microbiology. Rather than linking to outside videos to watch them within this, you'll be able to link to these outside websites and be able to research the information on your own as you have interest in any of these particular organisms. What we're covering is just a really basic overview of what these different organisms are. There is not a whole lot of video information out there that really works well for this class at this time. So a lot of the links are going to be from different online textbooks and other websites that have background information on these organisms that you can go ahead and follow. The links are going to be embedded right in the lecture material, so if there's something that strikes your interest as you're going along, you won't have to look too hard to find the information. The prokaryotes are the most diverse of all the groups of organisms. The morphology or shape varies greatly. The cocci are spherical organisms, with some being more oval and some nearly kidney-shaped and some pointed. The bacilli are rod-shaped, with some being pointed, some spindle-like, and others thread-like. They may also be spiral-shaped, with spirals being stiff spirals and spirochetes being flexible spirals. The vibrios have a slightly curved shape. A coccobacillus is an intermediate shape between cocci and bacillus. Some are pleomorphic, varying in shape and size. Others are star shapes, triangles, or squares. The most common asexual reproduction is binary fission. This begins with the cell replicating the DNA and each DNA attaching to the cytoplasmic membrane. As the cell grows, it moves the daughter cells and the DNA apart. A crosswall forms between the cell and evaginates the membrane. Eventually, the crosswall completely divides the daughter cells. The cells may or may not separate, but the parental cell becomes 2 progeny cells. Snapping division occurs in some gram-positive cells, where only the inner portion of the cell wall is deposited across the dividing cell. This puts tension on the outer layer that eventually breaks with snapping division. Some organisms form spores, which are reproductive cells at the end of a filamentous cell that can develop into a clone of the original organism. With budding, there is an outgrowth of the original cell. It receives a copy of the genetic material and enlarges until it is eventually cut off from the parent cell. The arrangement of the cells is the result of the planes of cell division and whether or not the cells separate. Diplococci are cocci attached in pairs, while the streptococci are long chains of cocci. Tetrads will form when the cells divide in 2 planes and remain attached. Sardinae divide in 3 planes and form cuboidal-shaped packets. Staphylococci are arranged in grape-like clusters, with the term staph meaning grape-like. When cells are in a side-by-side arrangement, they form palisades. Endospores may form in the gram-positive bacillus and clostridium. These are not reproductive, but a defensive strategy to ensure survival in unfavorable conditions. Endospores may remain viable for thousands of years. We have 3 main classifications or domains of organisms: the archaea, the bacteria or prokaryotes, and the eukaryotes. The archaea lack peptidoglycan in their cell walls. Their membrane lipids have branched hydrocarbon chains. The AUG codon codes for methionine, the same as in eukaryotes. There are 2 phyla of archaea, the Crenarchaeata and the Euryarchaeata. Reproduction may occur by binary fission, budding, or fragmentation. Many archaea are extremophiles, which require extreme temperatures, pH, or salinity. The thermophiles require a temperature over 45 degrees Celsius, and the hypothermophiles need over 80 degrees Celsius. Acidarius pyrodictium and Cephalobus are both found in sulfur hot springs. The halophiles live in extremely saline habitats such as the Dead Sea, the Great Salt Lake, and solar evaporation ponds. They require a concentration of sodium chloride greater than 9% or a 1.5 molar solution. Most require between 17 to 23% sodium chloride. Some will contain red-orange pigments. One example is the Halobacterium salinarium. The methanogens are obligate anaerobic euryarchaea. They will convert carbon dioxide, hydrogen gas, and organic acids into methane. They are found in ponds, lakes, oceans, and the intestinal tracts of cows. Due to this organism, cows are the primary source of environmental methane, which is a significant greenhouse gas. This is also what makes swamp gas.